Hi, welcome back to this YouTube channel. Today we are going to continue with macromolecules and we are going to have a quick revision on protein. This is the monomer of protein and it is called as amino acid. Amino acid have alpha carbon, which is the center carbon, carboxyl group, and amino group as well as R group as the side chain. Each amino acid has different side chain. The R group can be vary in structure, charge, and affinity with water. When amino acid is dissolved in water, it dissociates to form Zwitter ion. Amino group is ionized into NH3, whereas acidic group is ionized into CO-. The acidic group is a proton donor, where the amine group is a proton acceptor. This is the polymer of protein. Protein is made up of amino acid and they are linked by peptide bond. The peptide bond link amino group from one amino acid to carboxyl group of another amino acid. In the formation of a protein, H2O is removed. However, if you break down proteins into its monomer, H2O will be added to it. The backbone of protein consists of amino and alpha carbon and the carboxyl end. The side chain of the amino acid is the R group. Amino acid is classified into 4 groups and it is based on different R groups. Amino acid can be classified into electrically charged side chain, where it can be basic or acidic. Acidic side chain is negatively charged, whereby basic side chain is positively charged. Polar side chain is hydrophilic. It usually contains OH or SH. Lastly, non-polar side chain is hydrophobic. It is usually CH3. This is the levels of organization in protein. It consists of primary structure, secondary structure, tertiary structure, and finally quaternary structure. Primary structure is the linear sequence of amino acid. So you can see that amino acid is binding to one another joined by peptide bond. And this sequence of amino acid is determined by genetic code in DNA molecules that is located in nucleus. Secondary structure, on the other hand, is segments of polypeptide chains that are either coiling, or folded. There are 2 types of secondary structure. The coil form is called alpha helix. The folded form is called beta pleated sheet. The coiling and folds are basically the results of hydrogen bond between carboxyl and the amino group of the polypeptide backbone. Next one is the tertiary structure. It is the overall 3D shape of polypeptide chain that results from interaction between the R groups. These interactions include hydrogen bond, ionic bond, hydrophobic interaction and van der Waals interaction, and finally, the strongest covalent bond that may be formed between the R groups is disulfide bridge. Finally, the quaternary structure of a protein is the result of two or more polypeptide chain forming a protein molecules. Example here is collagen. It is a fibrous protein and it consists of three polypeptide chain coiled like a rope. So we can see here the red one is the first polypeptide chain, the blue one is the second polypeptide chain, and the green one is the third polypeptide chain. So we have three polypeptide coiling like a rope. Another example of quaternary structure protein is hemoglobin. Hemoglobin is a globular protein, consists of 4 polypeptides. It consists of 2 alpha and 2 beta chain. So we can see here, this is alpha subunit and this is beta subunit. And we can see here that hemoglobin have heme group, and heme group is attached to non-protein compound, which is iron. To classify proteins, it can be based on structure, composition, and functions. Structural classification of proteins can be divided into two. It's either fibrous protein or globular protein. Example of fibrous protein is collagen. It consists of long parallel chain. Example of globular protein is enzyme. It is basically coiled and folded into globular shape. To classify protein based on its composition, we divide it into simple proteins and conjugated protein. Simple protein consists only amino acid and it do not contain any other substance. However, conjugated protein contain protein and non-protein material, which is the prosthetic group. Example of simple protein is antibodies, and example of conjugated proteins is hemoglobin. As we can see before, hemoglobin contains heme group, and inside heme group it contains iron, which is a non-protein material. Finally, we can classify protein based on its function. We have 6 functions in total. The first function is structural function. Example of protein is keratin. Keratin can be found in hair, nails, scales, and feathers. Next one, protein that is used in movement. Example is the actin and myosin that can be found in our muscle cells. For defense, we are familiar with antibodies. Next one is protein that is used as storage, like albumin that can be found in egg white. Signaling protein is another example that is insulin. It is secreted by pancreas. It promotes glucose uptake into the cell. Lastly, proteins can be used to catalyze reaction. Example is enzyme. Here given to you is amylase that can be found in saliva to digest carbohydrates. That's all for proteins. Thank you and goodbye.